So, I mean, the meeting, we try and have a mixture of what people can do to help themselves now, which we've just had a very good example of, and we like to highlight what's round the corner, what's coming up on in the future. So I'm just going to highlight a few areas of current LAM research here, and there's a lot of stuff I could say, but actually the things I've limited myself to really are the things that are going to impact upon patients within the next probably 2 to 5 years. I haven't got any sort of blue sky stem cell sort of stuff in here, although whilst that's exciting, I'm sort of limited myself to what's going to be applicable fairly soon. So I'm going to talk about sort of 3 main areas really. An animal model of LAM lung disease, which is something that the field's needed for a very long time now here. The main bulk of the talk is how we can improve treatment with rapamycin, and that's based really on understanding how LAM cells that cause the problem avoid being killed by rapamycin, and by understanding these processes we can actually improve treatments. I'm going to talk about biomarkers a little bit for improving clinical management and research. Obviously, we're in very exciting times here with advances in these areas, and I'm just mentioning everything we're doing because I'm very excited about that too, but I'm going to leave Aru and Debbie Clements to talk about that this afternoon, really. So this is a sort of overview of what's happening in other places, really. So the first thing I'm going to talk about is an animal model of LAM lungs, and whatever you think about animal experimentation on its in ethical grounds, virtually every major disease area tests their new drugs in an animal system. There's animal models for diabetes, for liver fibrosis, animal models for testing out how influenza affects the system. Virtually any disease you can think of, most of new drug research happens in an animal model. And for LAM, as you probably know, LAM occurs when there's a change in the TSC2 gene in LAM cells, and the LAM cells become transformed and they grow and migrate abnormally. Now, people have always tried to model LAM in animals by stopping the function of the TSC2 gene in mice and rats, and when they do that, they get abnormalities, but they don't get any form of lung disease. And this has been a real limitation. You just can't test drugs on the main organ. And over the last year or so, we've had what I consider to be a pretty major advance in the field from Vera Krimskaya's group in the University of Pennsylvania, who've described a mouse model for LAM. So this slide here is a section of a normal mouse lung, and you can see it's these little air spaces here with the cells around them. These are normal-looking alveoli, perfectly designed to absorb oxygen from the air in the spaces here into the blood vessels, which are in the little walls of these. What Dr. Krimskaya has now done is taken cells from other animals that have the TSC2 gene, the gene that's faulty in LAM, switched off completely. These cells are produced in other animals and then injected into the veins of mice. And like in the human disease, these cells, these mock LAM cells that she's created, migrate towards the lungs and start growing there. And this panel here, labeled TSC2 null, this is a mouse that these cells have been injected into, and you can see that this is different from the normal situation. There are these little nodules of cells here, fairly similar to human LAM nodules. The other thing you notice is that the lung architecture here, the air spaces are much bigger than in the normal lung, and so these are the two features of the hallmarks of LAM, really: a build-up of LAM cells in the lungs and airspace enlargement, limiting oxygen diffusion. And the model, I don't know how well you're going to see this actually, the model also has various other features. This is human LAM tissue stained for various characteristics of LAM. So the green staining here Each of these little blue dots is a cell. The green staining is staining for this protein smooth muscle actin. This is the red staining here is for activation of the mTOR pathway, and these two things are lymphatic markers. And you see they're all present in human LAM nodules, and then the panels here are the mouse model. And in the LAM mouse model, in this central panel here, all of these features are also expressed. So the model very closely mimics the features of disease. And so this is an advance in as much as, you know, if we have an idea of what might be helpful for LAM, we can test it in a cell. But of course, one single cell is going to react in a certain way. But of course, a human with LAM has lots of other normal cells around, and the different interactions just can't be modelled. But actually using a whole organism is a much more reliable way of doing this. And this model is being currently used to understand the pathology of LAM and to test new drugs to treat the disease. And by way of following on from this, I'm now going to start talking about how experiments like this are currently trying to improve treatment with a standard treatment for LAM now, which is rapamycin. So the first study leads on from the mouse model, actually, and it's the idea that adding statins into treatment of LAM with rapamycin would give better treatment alone than rapamycin alone. And you have to understand a little bit about what happens in a LAM cell to work out how this works. So what you see, this protein called mTOR here is present in all cells, and it's an important switch. And when it's turned on, the cells grow. They have all sorts of mechanisms to survive stressful circumstances, and they migrate around more. Now, mostly in normal cells, mTOR is switched off all the time. You don't want cells growing in the wrong place at the wrong time. And the thing that switches mTOR off is this combination of two proteins, TSC1 and TSC2, and these are the genes that become abnormal in LAM. So if you have LAM, this off switch doesn't work. mTOR is switched on all of the time, and these proteins become activated downstream of mTOR, cells grow, and they have all these techniques to survive in stressful circumstances, and eventually the cells grow in the wrong places and start damaging the lung tissue. When you use an mTOR inhibitor, a drug like rapamycin, you only block part of this pathway. So this little box here shows what rapamycin blocks, and it's downstream of mTOR, and it switches off cell growth. So the cells stop growing, but what you can see here is that the survival pathway is still working. And so the cells stop growing, but they don't die. And so when you stop taking rapamycin, the cells start growing again, your lung function starts declining again. So rapamycin is mostly a lifelong treatment at the present time. So what Dr. Krymskaja has done in her work using this animal model is found another drug that inhibits the other side of the pathway. And this drug's actually simvastatin, a cholesterol-lowering drug, but by virtually a side effect of that, she's shown that it also inhibits proteins called Rho GTPases that affect cell survival. And when she treats cells or the mouse model with rapamycin and simvastatin, she gets a much better effect on lung damage even after the treatment's been stopped than when she just uses rapamycin alone. We see this is a mouse, and women with LAM are not mice, as we all know, and human trials are essential to show if this is a safe treatment. The good news about this study, of course, is that simvastatin is a very commonly used drug worldwide. There's lots of safety data. Millions of people across the world take simvastatin for raised cholesterol. And so if it's shown to be effective, then it'll be something that we can bring to the clinic really quite quickly. So Dr. Krymskaja is currently arranging the first study to see if adding simvastatin to treatment with rapamycin for women with LAM, not mice, will improve outcome. The Another area where people are trying to work out how to kill off LAM cells rather than just stop them from growing is by a process called autophagy. And LAM cells do a curious thing. When you stop them growing with an mTOR inhibitor, they start eating parts of themselves to survive. I mean, analogous to, you know, if you had a remote house in the mountains, you had a very long winter and got snowed in, ran out of fuel, you'd probably burn the furniture to survive. And it's the same sort of thing for the cell. They have no nutrients. Now stopped growing, their mTOR that they depend on is switched off when they have rapamycin. So they have to do things to survive. And the thing they do is they start using up bits of their own structure as nutrients. So, as I said, rapamycin just stops growth, but doesn't kill the cells. And it's this autophagy that LAM cells use to survive in times of stress. mTOR controls autophagy in these cells, and by blocking mTOR, you actually sort of help autophagy to some degree. And so LAM cells are in a sort of delicate balance between autophagy and death. And Lisa Henske, who's one of the leading LAM researchers who's at Harvard now, has been understanding the process autophagy in LAM cells and is using drugs that inhibit autophagy in combination with rapamycin to see if she can get better outcomes for patients. And there's another, again, existing drug that blocks autophagy called hydroxychloroquine. And Lisa's shown that in combination with rapamycin in models, those two drugs are much more effective at killing LAM cells than rapamycin alone. And Lisa is just setting up a study at Harvard treating women with LAM with rapamycin plus hydroxychloroquine to see if it's safe and to see if this gives any encouraging results. The third area that we've sort of dabbled with for a long time is estrogen. Estrogen is a sort of elephant in the room of LAM, really. LAM's a disease of women. It's clearly something that's important in a disease. If you give people more estrogen, such as with hormone replacement, LAM seems to get worse. But if you block estrogen on its own, it doesn't seem to improve the disease to any significant degree. And just recently, there's been quite a lot more interest in how estrogen and mTOR act together. And there have been a few papers in some very heavyweight scientific journals over the last year or two, again from the Henske lab and other collaborators, showing that blocking estrogen and mTOR, or just blocking estrogen more effectively, might be useful treatments for LAM. So, and the upshot of all these interesting approaches to see how actually killing LAM cells rather than stopping them growing is turning into clinical trials that are actually sort of either just getting off the ground now or actually happening. So there's all these trials have got funny names, which I can't remember what they really mean. But the SAIL trials testing chloroquine and rapamycin to look at inhibiting autophagy with Lisa Henske. The SOS trial is presumably simvastatin or sirolimus or something, I'm guessing, that Dr. Kremskaya's doing. That's starting soon. She's going through the approval processes. Blocking estrogen in postmenopausal women with letrozole is a study that Dr. McCormack's actually finished recruiting now. I think the patients are still going through the protocol. And I haven't had time to talk about these, but there are two other trials of the new kinase inhibitors at different sites. So there's a lot of new interest in improving rapamycin treatment, and actually things that real patients can receive in the context of a clinical trial at the present time. These studies, I think, these are early safety proof of concept type studies, and they need bigger studies before they became a treatment. But I think if none of these things are particularly far away, as these are all existing drugs. So all of these things are happening in the States. As you no doubt remember, we do clinical trials in the UK. Here's some evidence of various people collecting their tablets and doing their lung function in our studies in the past. And I would hope that when we come to evaluate these newer treatments, that they're going to take bigger studies, and we'll be able to bring these to patients here too. So the final area I'm just going to briefly touch upon is the area of biomarkers. Biomarkers are very topical in most diseases now. And the reason they're important is because they help clinical care, they make research better, they make it faster and therefore cheaper, and they can bring advances in studies more rapidly, and they can help clinical medicine. So a biomarker is a factor that tells you about whether a disease is present or not, or maybe how severe the disease is, or perhaps what type of disease you have, or it predicts a process. So, for example, you might want to do a blood test that tells you that you have a particularly active form of the disease, and then you might treat that patient differently. So lots of diseases have biomarkers. If you have a heart attack, you go into hospital with chest pain, you have a blood test to measure your a protein that comes out of damaged heart muscle cells. That's a biomarker that tells you you've had a heart attack. The main biomarker we've talked about for LAM in the past is a protein called vascular endothelial growth factor type D, or VEGF-D. A group in Japan and then the States have described that VEGF-D can be used as a diagnostic test, and we showed that if you use it in conjunction with the ARS guidelines, that most of patients with LAM can be diagnosed avoiding a lung biopsy. And so VEGF-D has been around for quite a while. The more recent development is a study by Lisa Young, who works with Frank McCormack previously in Cincinnati. And they have shown that VEGF-D might be a predictor of activity of LAM. So this is the main slide from their paper, which was published in the Lancet Respiratory Medicine last year. And so this is VEGF-D along this way. So the further you are towards the 10, the higher your VEGF-D level. And this is change in FEV1 over a year. So this is a measure of your lung function. And if you're below this line, then your lung function's getting worse over a year. If you're on the line, it's not changing. If you're above, it's getting better. And you can see that the patients with the lower levels of VEGF-D here are fairly close to the line, but as your VEGF-D gets higher, you're more likely to have a decline in lung function. I mean, there's a fair scatter around these points, but it suggests that VEGF-D might predict activity. Now, that would be really handy. The first time I saw a patient, if I could say, okay, your VEGF-D is very high, I'm more worried about you, I'm going to follow you up more quickly, or maybe I'm going to treat you with rapamycin much sooner than I would do if your VEGF-D is low. That means perhaps we could prevent lung damage happening before we'd made a decision on rapamycin. We could plan care much more effectively and perhaps prevent disability with these sorts of things. VEGF-D looks like a useful biomarker. There are other proteins that people are interested in biomarkers, and there's a lot of work going on in this area. We're interested in this protein cathepsin K, which is raised in the blood of only some women with LAM, which we think might be important. And a lot of groups, including the Americans and ourselves, are using techniques called proteomics to see what proteins are different in the blood of women with LAM, and use these as biomarkers. So we hope this approach should make diagnosis easier. If they can a marker of disease activity, then they might be suppressed by tablets used to treat LAM reasonably quickly. So we could see if a drug was effective within a short period of time if we had a biomarker. At the moment, we have to wait a year to see if your lung function's changed. If we had a biomarker that we could measure after a month or two, which these things might do, then we could make screening clinical trials much quicker and cheaper, and move things forward more quickly. And as I said, we might be able to predict who to treat earlier. So I think this is a very exciting time for LAM research. Jan said in her talk that Frank McCormack said that the speed of progress in LAM rivaled actually any other disease, and I completely agree the way things are changing. And a lot of these advances, I think, will be able to offer to people, you know, within a realistic timescale. It's not just what might happen 20 years down the line. So, in summary, then, we've had a lot of different areas that are going to accelerate discovery. So we were in this difficult period where you had an idea, you tested it in a cell, and then you had to make this great big leap of faith to going into a clinical trial with patients and lots of money and years of treatment before it worked. This was very slow. Having this animal model we can screen out the ideas to see if they're, you know, more robust. We can also use these systems to sort of look at and screen for other things that might be abnormal. So we can, when we go to a clinical trial, we're more confident, and we hope that biomarkers can make the clinical trials quicker and smaller and cheaper, so we can get to treatments a bit faster. So I think that's where we are with LAM research at the moment. I think it's an exciting time. There's a lot going on.